Okay, so for today, got some reminders for you. Some important information here. Monday, we're going to be doing a work day for the final project, and this is important to make sure that you are all thinking about some of your ideas. So I encourage you to do that before you come to class on Monday. Start thinking about what you're interested in, maybe some potential ways you would convey that, who you might want to work with, etc. I am going to send an email reminder probably today telling people that, because of course, if there's a lot of students who are missing, this is a much less effective use of time. So I do encourage you, if you have friends in the class who you're hoping to work with, tell them to show up, tell them to come. It will be important, and I will ask you to decide on your groups on Monday. So be thinking about that. There is going to be another work day. I don't know the exact day. It's next week at some point. Not next week, it's the following week. Last week of classes. About 3 would be good. If you want to do 4, that's okay. If you want to just have a partner, that's okay. But anything bigger than that is probably a bit big, unless you have a really strong reason for what each person's going to do. I'm fairly flexible, but aim for about 3 or 4. Yeah, any other questions on that? Cool. Kind of related, I am going to post a link to a lecture recording on speciation and phylogenies. So look for that. Please review that. The content in that lecture will be on the exam. I think it's a better use of time for you all to be together in class, able to talk about your final project and work on your ideas, rather than just a lecture. So again, if you have questions, please let me know, especially with that content that I'm not going to be lecturing about in person. Another reminder, I told you both of these things previously. Exam 3 is going to be May 5th, and then your final project is May 9th.
So Friday, next Friday, that'll be like a review.
Next Friday, let me look at a calendar. My brain's getting all jumbled. Whatever the day is before the exam will be a review, if that's the question. So May 5th is a Monday, yes. So Friday the 2nd would be a review day. Yep. Good question. Any other questions? Cool. And just a reminder, the 3rd exam, it's not a final exam. We're just covering the topics since exam 2. I do actually think I'm going to put, rather than bonus questions relating to the content from this exam, I'm going to put a bonus question section that are cumulative. So probably 5 to 8 questions from content throughout the semester. So if you remember some of those things, that's a great place to earn extra points. If you don't, it will probably be fine. I don't know yet. It depends on how hard the questions are and how many points the exam has in other areas. Anything else? Okay, today we're going to be talking about species definitions, taxonomy, and we might get into speciation a little bit, I'm hoping. If not, I do have that in your lecture recording. So there's no perfect analog in the book if you are following along. There are some components in the book chapters that you can refer to if you'd like. Our learning objectives for today, I want you to be able to define a species. We're going to talk about why the taxonomic system is important, why biologists use it, classify or use an example of the taxonomic hierarchy classification system, naming system conventions. We're going to talk about morphology and evolution. It's a pretty critical backbone to understanding taxonomy. We're going to also talk about modifications to traditional morphological taxonomic techniques as well. And finally, that speciation learning objective is describing the origin of new species and essentially the diversification. So when you look at this picture, what are you noticing here? What's in this picture? What are you seeing? Water. Okay, what did you say, Faye? Animals. Yep, we got a lot of different animals here. Like what? Yeah, frog, of course. Lovely. A couple of frogs. What else? Snails. Yep. Yep, there's a diving beetle. Yeah, it's a really cool pond. I think this is some sort of like weird illustration for aquatic ecology. But yeah, we're noticing lots of different things. Maybe we see this newt, what looks to be also a different type of newt. We're seeing some larvae. So these are eggs. And then we have like dragonfly larvae, a toad up here, dragonflies, etc. So the things that you all were naming in this picture, although you weren't using like a scientific name, you were mostly talking about species or specific groups of organisms, right? So this picture is just a very, very small representation of some of the millions of species that live on Earth today. We know the natural world is complex, and biologists work to solve problems, right? And essentially, we attempt to organize and categorize the natural world in order to better understand the relationships between organisms and to be able to provide protections. So taxonomy is essentially how we make sense of this picture, and one of the most important kind of fundamental units of organization in biology is the species definition. So species are defined and differentiated by their physical characteristics, their looks, and their relationships to each other. And we actually have a huge number of different species concepts. On this slide, I have 3 of the most common or most widely used species concepts. The first is morphological species concept, and this idea says organisms that share measurable traits are species. That's what this concept says. Species are defined as those that have differences in a physical appearance, and for those of you who don't know what the word morphology means, it refers to structures and characteristics that can be measured and observed. Now, some benefits of the morphological species concept. We can actually apply this to extinct species. So this is widely used with people who study things like paleontology, because again, we can't see if dinosaurs were mating or interacting in a certain way because they're not live anymore, but we can make measurements and compare them. So this can be helpful in some ways. Biological species concept is one you may or may not be familiar with, but this is a really prevalent species concept. This essentially says that groups of interbreeding populations that are reproductively isolated from other groups are species. Now, this idea of reproductive isolation essentially means that if they reproduce with a different organism, that will not have fertile offspring. So it's kind of tricky, and I'm going to talk a little bit more about this definition in today's lecture. Now, lastly, a common species concept you might come across is the phylogenetic species concept. And this defines species as the smallest group of organisms that share an ancestor, so a common ancestor within their evolutionary history, and that can be distinguished from other groups of organisms, again, by their evolutionary relationships. Now, what's cool about phylogenetic species concepts is it's using both morphology and genetic information. So phylogenetic species concepts are actually really thorough and very, I don't know if accurate's the right word, but they do a good job of distinguishing species. They can also include both asexually reproducing species and extinct species. When we think about the biological species concept, we're like, okay, if they can interbreed, then they're a species. But that leaves out asexually reproducing organisms, right? Because they're not interbreeding with other members. They're just reproducing basically in a cloning situation. Same deal with the extinct species. We can't see if they're interbreeding because they're not going to be alive. So the one reason that the phylogenetic species concept isn't super duper widely used is largely because we don't have the full extent of evolutionary history for all of the organisms on the planet. In fact, we don't have anywhere close to that. So this is kind of a development that we're looking into. Questions on this before I move on? Okay. So this is a figure that I grabbed from a paper that was looking at differences. It was a review paper on different species concepts. Now, you'll notice here there's a whole lot more than what I was just talking about on the previous slide. So you can see in this column here, we have all these different species concepts. And if you wanted to, you could go read about more of these. Some of these are kind of specific, some are a little bit more broadly applied. But something that's kind of interesting is the fact that some species concepts are incompatible, which means they're going to come to really different conclusions about the number and definitions of species that exist. So species concepts are kind of funny because essentially they're a human imposed idea on this like complex natural world, right? So it's just our way of trying to understand the complexity that we observe, but there's no real right answer to this. That's why it's kind of so variable, essentially. Now, getting a little bit more focused here, as I was saying, this idea behind defining species is how we're trying to organize the natural world. It's the most fundamental grouping. There are things called subspecies, which are technically like a little bit more specific, but we're not going to be focusing on that in this course. And some scientists don't really like to recognize subspecies, so it's a little bit controversial as well. In this class, we're going to use a biological definition of species, so a group of populations in which individuals are so similar in structure, biochemistry, and behavior, they can successfully interbreed. So when they breed with each other, they have offspring that are fertile. They can successfully interbreed. That's our definition. Now, according to most species concepts, or what most scientists tend to accept, we have about 2.16 million described species. Now, described means they've been recognized, they've been studied, they've been named within our scientific system. However, this is definitely not the total number of species that exists on the planet. And also, most of the time, we're kind of just guessing with these estimations. We think there's about 5 to 10 million eukaryotes. That's a relatively up-to-date number. Now, eukaryotes, of course, do not include things like archaea and bacteria, of which we cannot even begin to estimate the number of those organisms, but it's millions for sure, maybe even billions. So it's kind of interesting if you spend time in the literature on the number of species that exist. Some people say, okay, we could defend anywhere from like 3 million to like 1 billion different species, which is a huge difference if you think about that in terms of the number of species. Now, some people say, like, why bother? Why should we care looking at this or learning about this? There is a strong argument in conservation biology, essentially, that species are going extinct before we even know they exist, which is a huge problem, basically because we can't catalog the biodiversity on life and on Earth and the biodiversity that exists, which is challenging. So if you want to talk more about this, I'm happy to. And we're not going to talk about human impacts or conservation biology in this course, but we do have other courses in the department that go into it. And it's also what I like to talk about a lot. So feel free to come chat. Now, as I was saying with our biological species concept, these designations rely on reproductive compatibility, or the ability to reproduce, and that leads to these things we refer to as isolating mechanisms between different species. So we have two different types of isolating barriers or mechanisms, and one is our prezygotic isolating barriers. So break that word down. What does prezygotic probably mean? Yeah, before zygote, absolutely. And what's the zygote? Before an embryo, that's true. Yep. If we were to be a bit more specific, it's a combination of two things. Yeah, it's the fertilized egg. Yeah, and there's two haploid cells combining to form that fertilized egg. So the zygote is the fertilized egg. So prezygotic isolating barriers are essentially factors that prevent the production of hybrid offspring in the first place. These could be a lot of different things, and I have a slide going into more detail on some of these ones that I've listed here. Habitat, temporal, behavioral, mechanical, or gametic. So for example, habitat would be like if two different species lived in different areas, they're not going to reproduce because they're isolated geographically. That makes perfect sense. Other things like behavior, maybe one species likes to mate in the morning and the other mates at night. That would also be a prezygotic isolating barrier. They're just simply not going to have compatibility in that area. Now, post-zygotic isolating barriers are if those organisms actually do have a mating event, there's other ways that are going to essentially reduce the fitness of their offspring. So sometimes there are closely related species that do interbreed, but most of the time the embryos die early on. Sometimes those offspring, if they do get if they develop and they come to fruition, sometimes later on in their life they're sterile, or over time they don't live as long, or they have reduced fitness themselves. So, questions here? Mm-hmm.
Sort of stuff, then, like, sort of, to make different species.
Yeah, is it ethical to have these mating studies? That's a good question. So it's definitely an area of research, and it's not so much that we see mating studies as much as scientists tend to observe what happens in the wild. A lot of us like to maybe assume, or we don't think about it very much, the fact that in nature there's lots of hybridization, actually. We see lots of fairly similar species actually interbreeding. And so speciation and evolution is kind of always happening. And most of the time they're studying what happens in nature. Sometimes they do things like fertilize eggs in the lab, and that is relatively, I don't know that anybody has ethical issues with that. It's more so once those individuals would potentially be like growing up in harmful scenarios or if they had incompatible features. But yeah, that's a good question about the ethics of that. Any other questions here? And like I said, I have more details on some of these factors in a couple of slides. So what happens if two unrelated species mate? Some of you might know what some of these organisms are. Does anyone know what's on the slide? Yep, a mule. So a donkey and a horse. Okay, what about this one? This is a liger down here. Goat is a good guess. Ox is a good guess. It's actually a bison. So this is a beefalo, right? This is classic. Over here, most monstrous of all, the Savannah cat. Okay, so all of these are hybrids, which is the definition of two unrelated, or not unrelated, two separate species reproducing. That's what hybrids are. Male ligers tend to be sterile, whereas female ligers can actually tend to reproduce with other either lions or tigers. Now, this is kind of interesting, and if you want to read more about it, you definitely can. The heterogametic sex is more likely to be affected by hybrid issues like sterility. So heterogametic, remember, refers to having two different sex chromosomes. So depending on the species, sometimes that's the male, sometimes that's the female. But with a lot of mammals, we know that the female tends to have those two X chromosomes. So there's less problems with their reproduction in these cases of hybridization. Now, savanna cats are a cross between domestic cats and wild African servals, and they're very popular as pets, which is kind of crazy. Beefalo can actually produce offspring, and I'm pretty sure they're now recognized by the USDA as actually like their own species. Now, this is kind of interesting as well. So quite a while ago, I think in the early 20th century, maybe the 19th century, bison were pretty much eradicated in most of their native range, which was really problematic in terms of genetic diversity. So over time, some folks thought it would be a good idea to kind of mix bison with cattle to provide some of the benefits of wild bison, along with some of the benefits of like more easy to handle cattle. Which I mean, some ways was successful, but we know that they're also more susceptible to both like bovine diseases as well as diseases that affect wild bison. So that's kind of an interesting area to explore as well. They're called like beefalo, but colloquially, I'm not sure that they have a scientific name. And I'm pretty sure actually the bison out at Frontier Park are beefalo, actually. I think they can breed with like other cows, but it's hard to find information actually like that's super, super clear. But I'm pretty sure they can either breed with other cows or other beefalo. Someone else might know. Okay, so I have a question here for you. Given what you've learned about meiosis and reproduction, I want you to chat with someone near you about why hybrids are often sterile or have issues with sterility. So think about what meiosis does, what is that process, and what might be impacted causing this issue. Thoughts here? Do y'all have an answer or an idea? No. Does anyone have a guess? Let's back it up here. Okay, what does meiosis do? Produces gametes, yes, okay. So gametes are really critical. They're the reproductive cells, right? And so, yes, meiosis produces gametes. And oftentimes, the reason that hybrids are sterile is largely because actually that meiotic process in those hybrid individuals is not successful. So they oftentimes can't really even produce those gametes, largely as a result of like chromosomal incompatibilities. And the different hybrids have different issues. So it's not like across the board we see the same thing, but generally there's meiotic failure to produce gametes is why they're sterile. Questions on that? Mm-hmm. Could there be a mutation that makes them sterile? Yeah, in theory, there could be. Usually that's not what the problem is. I would actually, we could kind of apply like an interesting evolutionary theory to that idea. If there were species that were constantly mating with each other and we were seeing a lot of hybrids, over time we would actually expect maybe there to be some version of a, maybe not a mutation, but we might expect their over evolutionary history to be able to accommodate that, and we might see changes. I don't know if it'd be a mutation, but maybe they would kind of converge into a single species over time. Do you have a question? Like always, forever? Yeah, so like every time that we like see matings between donkeys and horses, for example, they're always sterile. And most of these animals are pretty much created by humans. So they're at very different like artificial selection and selective breeding pressures. Whereas in wild populations, typically hybridization occurs between really, really closely related organisms, things we call sister species. So a good example is with like frogs. A lot of times there's sister species that look almost identical, and the only difference is maybe like the number of chromosomes they have. Over time, we would expect there to be potentially some change because there is a fitness effect of hybridization, right? It's costly to mate, and we know that evolutionary pressures favor fitness benefits or organisms that have higher fitness. So things that reduce your fitness, we would expect to be under evolutionary pressure. So it is possible that it would change over history. In the cases of things that like we're making, for example, domestic cats with wild cats, probably not, but maybe. Good questions. And speaking of that, some people are surprised to hear about savanna cats. I feel like they show up on the internet a lot, and it's valuable to kind of talk about them. So has anyone heard of Bengal cats or seen cats that look like this? Okay, so Bengal cats are another popular pet breed. They're mixed with Asian leopard cats and domestic cats. So wild cats from Asia, they take them from Asia and then they breed them with domestic cats and then they sell them to you as a lovely pet, the Bengal. I think they cost like $1,000 to $2,500 for a Bengal cat. We also have the Savannah cat, which you can see here next to the counter. Savannah cats are really large, and this picture previously, this girl's holding them. These are kittens, so these are not even full grown, and they're really quite large. They are a mix, again, between domestic cats and African serval cats. So they're, of course, maybe you're wondering, why would people do that? Maybe you're thinking, that's cool, I should buy one. First of all, no, don't do that. Probably a bad idea. We know that most of the time they retain a lot of wild behaviors and wild characteristics. So things like really high energy, sometimes they're very aggressive. They can be very territorial. Most people don't like cat pee in their house. I don't know anyone who likes it actually. And these cats tend to show those behaviors more often than just domestic cats. We also know, and you might have heard of this, especially if you're familiar with like the feral cat population or the wild cat population in Hayes. It's a big problem. And one of the reasons why is because cats are excellent hunters. Domestic cats, like just pure domestic cats, are great at catching birds, catching snakes, catching mice, catching mammals. And they do serious ecological damage, like big harm, which is why you should keep your cats inside. But we know that when domestic cats are actually hybridized with wild cats, they're even better hunters. And those genes can actually get mixed into populations of wild cats and domestic cats as well. So we see issues with both the continuance of these species from a pet trade component, from a hybrid risk component, and we know that these types of pets are really, really damaging to the environment, and they're expensive, and they're not really good pets. So this is kind of hybrid cats. I don't know why they started doing this. They should stop doing this, but yeah. Say what? Yeah, you shouldn't. I actually do have a friend who got a Bengal cat, and it's crazy. They should have asked my opinion before they did it. Yeah. Yeah. No, that's a good question. Yeah, farm dogs are, because I'm a cyclist, my nemesis, to be honest. They chase me all the time. And they're very effective because most of the time dogs have a high protective, especially like the ones that are used as farm dogs, like Great Pyrenees. They're very protective of their owners, which is why they make good protectors. Cats do have like relationships with humans and they do like humans, they like companionship, but they definitely probably wouldn't be super good at warding off like a bear. They would get eaten by a coyote for sure. Yeah, an African serval could probably fight off a coyote, but this guy is still half domestic cat, you know? Domestic cats versus coyote, no good. Raccoons even can do some damage. Servals? No, most wild cats are pretty lone like animals. They have like a home range. Yep. Anything else? My weird hybrid cat diatribe? No, no, I found that online. I don't know anyone with a Savannah cat. Those are seriously crazy. If you look up videos on like YouTube, for example, you can Google like Savannah cat taking whole chicken off the counter, and they're just like, "Ah," like give me the meat. They do growl. Yeah, they're very aggressive. They want that flesh, you know? Can't blame them. Okay, going back into some more of the specific details here, as promised, prezygotic barriers I'm going to detail here. So habitat, again, two species that might occupy different habitats in the same area, they're not going to encounter each other. That's just because they have these specific niches, these areas where they occur. This is an effective prezygotic barrier. Temporal, I was also kind of talking about this. They might breed during different times of the day. This could actually extend into other areas, maybe different seasons, spring versus fall, or even different years. We know some organisms only have mating events or breeding events on like not annual cycles, but within like a yearly cycle. So this can be a barrier as well. Behavioral is kind of cool if we think about it. And I was kind of talking about last class with birds. There's a lot of like sexual selection factors, things like dances and nest building, different colorful feathers or calls, things like that. Courtship rituals for mates are usually very unique, and those can be good isolating factors. With frogs, that would be like their call. That would be a behavioral courtship ritual. That's how they attract mates. Longer, louder calls indicate, better mating opportunities. Mechanical mating barriers are essentially where the two species might try to mate, but they won't be able to simply because there's like physical issues with that process occurring. There's morphological differences that prohibit the successful completion of that mating event. And then gametic barriers are those where like sperm from one species cannot fertilize the egg of another. This is largely due to the fact that sometimes the reproductive tract might be inhospitable in the female, for example. We also know that sperm and egg have recognition systems. So if the egg doesn't recognize the sperm, aka if it's from a different species, most of the time it's not going to let that sperm in to fertilize, which is kind of cool that that exists. But we see lots of different prezygotic barriers. So this is an example of a mechanical isolating barrier. Now here we have some snails in the genus Bradybana. Snails are interesting. You probably don't know this. This is a fun fact. So snails have different chirality, which refers to the direction that their shell swirls. So their spiral is referred to as their chirality. And snails actually can only mate if they have the same swirl direction, if they have the same chirality. So the arrows here are showing the genital openings that have to be aligned in order for these snails to reproduce successfully. So you'll notice these two snails are able to successfully mate because they have the same chirality, whereas these two, it's on the wrong side. So they have to line up like that, and right now they're lining up the wrong direction. So kind of funky. Snails are really weird. They also can be hermaphroditic, and they have lots of other interesting reproductive components. So it doesn't work. I don't know why they couldn't just roll over, but the way that they have to actually interact, like the genitalia, it doesn't work. Which is weird. Chirality is the thing. Kind of crazy, I think, anyways. hmm Just looks like it just rolled over. Well, it does kind of seem like this one's a little bit like pushing it the other direction, but they're definitely interlocking in this picture. Maybe I should get some more zoomed in pictures of the specific snail genitalia in the future. Okay, postzygotic barriers. Remember, postzygotic is after the fertilization of the egg has occurred. Reduced hybrid viability. Essentially, the genes of the two parent species interact in ways that are disadvantageous, and the survival of the offspring is not benefited in its given environment. So, postzygotic barriers overall prevent a hybrid zygote from developing into a viable, fertile adult. Reduced hybrid fertility is where if the chromosomes of two parent species differ in number, meiosis may not produce normal gametes. This is what we most often see with hybrids. This is what I was talking about on that slide with the mule and the liger. And then hybrid breakdown is kind of interesting. If at first hybrids are fertile and viable, over time, as they continue to mate with the original parent species, sometimes future generations are impacted. So sometimes over time, we see this breakdown, so they're less successful. Questions here? Okay, I have a break, kind of an activity for you. With a partner or alone, I don't care. On a scratch piece of paper, I want you to illustrate gene flow. And importantly, do not use the internet and do not use your notes, just your brain and or the brain of your friends. Highlight relative influence of this process on the genetic variation in a population. So you don't have to go like really, really deep with this. Just a basic overview here. Just a quick little break. And you're not turning this in, so you don't have to worry about like getting a separate piece of paper. Nice. In terms of the other mechanisms, so the ones we talked about were natural selection, gene flow, and genetic drift were the main ones. I also talked about mutation as like sort of a additional mechanism for evolution. Yeah, gene flow. And like I said, you can use your friends to develop this idea or to develop your drawing. What's up, Jordan? You're looking at me. I don't know. Coy, Tyler, what do you think? Where the bugs get stepped on? Okay, so do you remember what genetic drift was? Do you remember when I was talking about genetic drift?
What I was thinking.
That's when something dies.
I'm sure, like where there's like a really random event potentially. Yeah, and what does the effect of genetic drift on a population? So yeah, if they're dying, we would expect usually like a reduction in genetic variability. Yeah. So then gene flow, do you remember? No, I don't think you're dumb. You're doing a good job. Without notes, that's tough.
I got the question you asked me.
About hybrids, and you didn't answer? Sorry. About gametes of hybrids? Yeah, you should always answer my questions loudly. You don't want to be wrong in public. Embarrass yourself. I had someone say one time that they're like, I like to answer your questions because even when I'm wrong, you make me feel like I'm right. So maybe you should try it. Maybe I'll.
What are your religious beliefs?
You have to come to my office hours to get answers to questions like that. Answer the question. How's it going? Oh my gosh, you're using notes. Oh my God.
I wasn't here when it said, so I didn't.
Okay, fine. Fair enough. I'm just kidding. Did you have it locked in?
Yeah, it was like a bunch of different color of the species, and then the other, and then the one,
Bugs all over.
then the one different color goes into the other color, and like this.
Uh-huh. And then what's the impact on genetic variation in a population?
It increases the variation.
Increases?
Population.
Yep. Okay, cool. Lovely. Awesome. Gene flow. How's it going over here?
Getting somewhere.
Okay, lovely.
Right? Like kind of.
Yeah, the arrow is gonna do a lot for my interpretation of this.
It's gonna have clear buggy.
The clear translucent bug joining the population of darker bugs.
Yeah.
Okay, cool. Love. Oh, say what?
Those are bugs.
Those are bugs. Okay. Love it. And what is the impact on genetic variation in the population? Say what?
Wouldn't it just it like increase?
Yeah, increases genetic variation. Cool. Love. Okay. Got it? Yes, with your math homework.
Fine.
Got it in my mind. Okay, so I talked to a bunch of you all. So gene flow, we can also kind of think about this as migration. So that can be helpful. Gene flow is when we have migration into a population. It's also really technically defined as the movement of individuals and or the movement of genetic material. So when we say gene flow, it's the movement of genetic material. Movement is like migration. Maybe you can remember it that way. I don't know. Gene flow can include a lot of different events. Sometimes that could even be like pollen being blown around. Maybe there's a person moving to a new city that could kind of be considered gene flow. Questions on this? Okay, we're going to keep going then into taxonomy. Yes, I did already show you this picture. We're coming back to it. So we're talking about different organisms, making sense of the complexity that exists in the biological world. And we use taxonomy to really do that. Taxonomy means arrangement law. That's what this translation of this word essentially means. And this is a whole branch of science. There are taxonomists. There's people who devote their whole careers to differentiating, splitting, and classifying species. I personally I'm not a taxonomist. I don't like it. I don't think it's my favorite use of time, but it is really important in understanding it. So this is kind of a good insight into all the different worlds of science. So I myself, I'm really interested in herpetology, but I more take a conservation or an ecological perspective, as in I'm most interested in what the organisms are doing in their environment, how they're impacted by humans and other organisms, whereas a taxonomist might be more interested in like how they differentiate from other species, what differentiates them, why they can't interact, etc. Now, the way taxonomy works is we have these kind of classifications or groupings that are increasingly more inclusive. So we've previously talked about the domains of life, and there's 3 domains, which would be our most broad categories, and then we get increasingly more specific. So when we look at this kind of like baby toy thing, we see like a broader category here, and then species as our most specific category. So do these figures make sense? They're kind of opposite. So you can see this one is saying like most broad to most specific, and this one is going the opposite way, most broad to most specific. Now, historically, taxonomists worked mostly using morphology. So remind me, what does morphology mean? Characteristics, yeah, is that what you're going to say? Yeah, awesome. So we're looking at specific measurable traits or characteristics, absolutely. Now, this is also sometimes known as the Linnaean system of classification. It's most inclusive to least, and organisms within any category of the hierarchy make up what we call a taxon. So you could say, oh, organisms are in this phyla. That's a taxon. So the plural for taxon is taxa, and you might hear those words used a lot. It's just a vocabulary term, essentially. Now, each of these taxons are defined by a certain set of shared characteristics, and I have an example for you in the next couple of slides. It's kind of silly. What makes the panda different from? They're herbivorous. Yeah, so they were looking at carnivora, so carnivorous organisms here, and then Ursidae. Oh, okay, I see what you're saying here. So these guys are not going to be eating a lot of meat, but I think over here, this is a tiger that we have, and that would be different. Ursidae refers to bears.
Why is it panda in the versus gene?
It is in the family.
No, in the gene.
Oh, now that I don't know. Probably their separation because they've been in Asia, and I think Ursa might only apply to bears that are in other parts. That's a good question. I actually don't know. I'll have to look into that. That's just a guess. Yeah, I'll have to make a note to look that up. Any other questions here? And this figure is kind of crusty because I photocopied it from a textbook because I liked it. So if you're wondering why it looks like that, that is why. Now, this is an important slide because I will probably ask you a question on the exam relating to scientific naming systems. So if you have questions about this, please let me know. Each organism is given a 2-part scientific name, which includes its genus and the species. Now, they have to be italicized. So if you're writing them correctly, they're going to be italicized. And you have to have the genus and the species. So it's only meaningful if we have the representation of both parts. However, a lot of times scientists abbreviate the genus. So in this example, I'm using the scientific name Crotalus horridus. And then in the second part, I'm saying, okay, we could abbreviate Crotalus horridus as just C. horridus. Does anyone know what that is, Crotalus horridus? I'd be shocked if you did. An isopod. It's not an isopod. It's a rattlesnake. It's a timber rattlesnake. Horridus. Ugh. Okay, why do biologists use this naming system with this specific two parts? Why do they use this system? Any thoughts here? Sure.
Like two of the hierarchy.
Yeah, it's the most specific components, absolutely. Now, when we think about a binomial nomenclature, that means naming system. And the thing on the slide, so I heard someone call it a roly-poly. I think someone called it an isopod. Does anyone have any other names for this? Roly-poly. Yeah, when I was a kid, I grew up in Utah and we called them potato bugs. So there's a lot of, yeah, I know. Apparently it's just a Utah thing. But there are a lot of different common names, so just colloquial names for different organisms. So this snake up here is a timber rattlesnake. This is Crotalus horridus. However, depending on where you go in the United States, you might hear this called a canebrake. It might be called a velvet tail. It might be called an eastern rattlesnake, or most confusingly, a black rattlesnake. Now, this picture is actually of a black-tailed rattlesnake, and then this is of an Arizona black rattlesnake. So you can see here, when we start using common names, it's not very effective at differentiating closely related species, because these are 3 different species of rattlesnake, which maybe they look really similar, but they have very different characteristics, temperament and habitat, location, prey specialization, etc. So the reason biologists use this naming system is because it helps us be specific and differentiate what we're talking about in really broad areas. So the biological naming system is used globally. Everyone, everywhere uses the same system, which is cool. We do actually have sort of a different system for birds. Most birds just use a common name, which is kind of funky. I don't know why. Ornithologists are just weird like that. Birds in general, so they do actually have scientific names, but they're mostly called by the common name, even within like scientific communities, which is kind of weird. Yeah, good question. Yeah, all of these are at the Sternberg. Yep, absolutely. So timber rattlesnakes, the one on the top, they live in Kansas in the eastern part of the state. And then these obviously live only in Arizona. But yeah, we do have some of those snakes at the Sternberg. No, you can't find timbers or any of those other ones on the trail. There would be prairie rattlesnakes, but not timber rattlesnakes. Okay, so I'm going to demonstrate using this system, and this is obviously kind of a silly example, but the idea here is to really kind of observe how this goes from really closely related or really similar characteristics to more broad. So here we have related species placed into a genus. So most of these snakes are in the genus Crotalus, which are vipers in the United States. However, we have this little dude down here, the pygmy rattlesnake. They're small, they're adorable, I love them. And they're in the genus Cisturus. And the only difference between Crotalus and Cisturus is the number of scales on the snake's head. Crotalus have lots of tiny scales and Cisturus have larger scales. So obviously not very much differentiation here. They are all venomous. Yep, so they're all vipers. Yep. Now we could place related genera into a family. And here we're looking at snakes in the family Viperidae. So Viperidae are all venomous. They all have keeled scales, which just means their scales look kind of like this. And they have a line, and that line is the keel. They have hollow fangs. They have vertical pupils. So here we can see still some similarities. However, these snakes that I was showing you before, our pygmy and our timber, we're seeing some differentiation here. This one is kind of weird. It has a different face shape. The eyelash viper has these distinctive modified eye scales. This, does anyone know what this is? They also live in Kansas. Snake, copperhead. Yes, no rattle on this one. And then this is a gaboon viper. They have really weird, big, puffy heads. So some differences here. Now we can put related families into an order, order Squamata. Now, Squamata is an order of scaled reptiles that have noteworthy jaw mobility. That's the characteristic. Noteworthy jaw mobility. So a good ability to move their jaw. That's their defining characteristic. Amphibians are the weird ones on the top. They're kind of like snakes in that they're scaled, but they're mostly fossorial, so they tend to live underground. Very interesting. Colubridae are non-venomous snakes. Does anyone know what this is? Komodo dragons. Yeah, venomous lizards. Okay, also a venomous lizard. This is a Gila monster. Which one? So varanid lizards, yeah, monitor lizards are in the group Varanidae. Yeah. Okay, so we could place related orders into a class. In this example, class Reptilia. We have Testudines, those are turtles. We have Squamata, Crocodilia, which I've talked about before. The lovely Gharial, my favorite of all the crocodilians. Now, Rhynchocephalia, this is a crazy organism. This is the only surviving species in its order. And they live in New Zealand. They're endemic to that island. And they look a lot like lizards, but they're really not lizards. They're very different. The Rhynchocephalia. Yeah, this dude. They look like lizards, but they're really not. I'm sure it's not a lizard. You should look them up, though. You should spend some time. Spend your time Googling. Huh? No, gharials are sweet angels. Kinda, yeah. There's actually not been documented crocodile deaths in the United States. That's interesting. Way more deaths by alligators. If you're looking for the common name, by the way, that's a tuatara. So Google tuatara. You'll have fun on that journey. The Rhynchocephalia, the specific name for that organism is a tuatara. You shouldn't buy them. They're critically endangered. Okay, reptiles are ectotherms. We're going to get bigger here. Related classes are placed into a phylum. So here we have chordates. We're all united by our backbone. So our spine, our ossified vertebral column. Shout out. We have some fish. Does anyone know what these are? They're gar. Yeah, long-nosed gar, also sweet baby angels. Emerald toucanet, a spring peeper, me and my cats when my cat was a kitten. We're all vertebrates. We're all chordates. Next up, we have phyla being placed into a kingdom. Now, kingdom Animalia is, of course, very broad. And when we look at this taxonomic level, we see very few similarities, right? If I was like, okay, tell me the similarity between an octopus, an atlas moth, and a segmented worm, what would you even say? They're all eukaryotes. Yeah, that's a great similarity. They're all animals. That's pretty much all we have here. So we have a lot of differences and far fewer similarities. Yeah.
What other kingdoms are there?
Kingdom Animalia. It's a good question. Plantae. I'd have to look. I'm not sure if they've changed that recently, but like Plantae is another kingdom. I think fungi might be in their own separate kingdom these days. Good question. Kingdom Protista, yes. Cool. So remember, we have 3 domains, which would be our most broad category. We have talked about the domains before: eukarya, bacteria, archaea, and all of the organisms I just showed you. Those are, of course, all in which domain? Eukarya. Yeah, cool. So I think we'll probably stop here. This is what your break was going to be. Is the ocean a soup? Defend your argument. But we only have 30 seconds left. So I'll get started with, yeah, I'll let you out 30 seconds early today. It's your Friday treat. Yeah, I'm very benevolent. I'm going to post a lecture. It'll go over taxonomy and phylogenetics. Make sure you watch it. The sound might be crappy. If you have questions, let me know.